Cell, the unit of life. Noteworthy points of the chapter. Cell is the fundamental structural and functional unit of all living organisms. Anton von Leeuwenhoek first saw and described a living cell, and Robert Brown discovered the nucleus. Schleiden and Schwann together formulated the cell theory. Omnis cellula e cellula was explained by Rudolf Virchow. Cells that have membrane-bound nuclei are called eukaryotic cells, while cells that lack a membrane-bound nuclei is a prokaryotic cell. Ribosomes are non-membrane-bound cell organelles found in all cells. The smallest cell is mycoplasma of 0.3 micrometer in length. The four basic shapes of bacteria are bacillus, rod-like, coccus, spherical, vibrio, comma-shaped, and spirillum, spiral. A typical eukaryotic cell is of 10 to 20 micrometer, and the size of viruses is 0.02 to 0.2 micrometer. Many bacteria have small circular DNA in addition to genomic DNA and is called plasmid, extra chromosomal DNA. In bacteria, the infoldings of cell membrane is called mesosome, which is the characteristic of prokaryotes. The cell envelope of prokaryotic cell consists of three layers: glycocalyx, cell wall, and plasma membrane. In some prokaryotes, glycocalyx could be loose sheath called the slime layer, and sometimes it also may be thick and tough called capsule. In bacteria, cell wall determines the shape of cell and provides structural support. In cyanobacteria, the membranous extensions into cytoplasm called chromatophores contain pigments. Motile bacteria have thin filamentous extension from their cell wall called flagella, which is composed of three parts: filament, hook, and basal body, in which filament is the longest portion and extend from the cell surface to the outside. Surface structure present in bacteria are flagella, pili, and fimbriae. Ribosomes are about 15 nanometer by 20 nanometer in size and are made up of two subunits. Prokaryotes have 70S ribosome, 50S plus 30S subunits, while eukaryotes have 80S ribosome, 60S plus 40S subunits. Several ribosomes may attach to a single mRNA and form a chain called polyribosome or polysome. Reserve material in prokaryotic cell are stored in cytoplasm in the form of inclusion bodies. The cell membrane is composed of lipids and are arranged in a bilayer. The membrane also contains protein and carbohydrates. In humans, the membrane of erythrocyte has approximately 52% proteins and 40% lipids. According to the fluid mosaic model of 1972, the quasi-fluid nature of lipid enables lateral movement of proteins within the overall bilayer. This model was given by Singer and Nicholson in 1972. Movement of water by diffusion across plasma membrane is called osmosis. Sodium-potassium pump is an example of active transport. The primary cell wall of a plant is capable of growth, and secondary cell wall is formed on the inner towards the membrane side of the cell. The middle lamella is a layer chiefly made up of calcium pectate, which glues neighboring cells together, and plasmodesmata connect the cytoplasm of the neighboring cells. The endomembrane system includes endoplasmic reticulum, Golgi complex, lysosomes, and vacuoles. I repeat, endoplasmic reticulum, Golgi complex, lysosome, and vacuoles. The endoplasmic reticulum bearing ribosomes on their surface is called RER. The SER, which does not have ribosomes on the surface, is a major site of synthesis of lipids. The Golgi cisternae are concentrically arranged near the nucleus with the distinct convex or cis phase or the forming phase and concave trans phase or the maturing phase. The vesicles from the ER fused with the cis phase of the Golgi complex and move towards the maturing or trans phase. Golgi apparatus is the most important site of formation of glycoprotein and glycolipids. The lysosomal vesicles are rich in hydrolytic enzymes and are optimally active at acidic pH. The vacuole is bounded by a single membrane called tonoplast. In amoeba, the contractile vacuole is important for excretion and in protist, food vacuoles are formed by engulfing food particles. Mitochondria and chloroplast are double membrane bound structures. The inner compartment of mitochondria is called matrix and possesses a single circular DNA molecule. The cell organelle mitochondria is the site of aerobic respiration and it divides by fission. Plastids are classified based on the pigments into chloroplast, chromoplast, and leucoplast. A myeloplast, a type of leucoplast, stores starch, carbohydrate. Elaioplasts store oils and fats, whereas uroplasts store proteins. Chlamydomonas, a green algae, has one chloroplast per cell. The space limited by the inner membrane of the chloroplast is called stroma. Thylakoids are arranged in stacks like piles of coins called grana and are connected to other thylakoids of different stacks by membranous tubules called stroma lamellae. The grana in the plastid is the site of light reactions and the stroma is of the dark reaction. The ribosomes of chloroplast and mitochondria are of 70S type. Ribosomes are composed of ribonucleic acid and proteins. Eukaryotic ribosomes are 80S type. Here, S is the Swedberg's unit, which stands for the sedimentation coefficient. The core of the eukaryotic flagella is called the exoneme, which consists of 9 pairs of doublets radially arranged peripheral microtubules and 1 pair of centrally located microtubules. The arrangement of microtubules in eukaryotic flagella is referred to as 9+2. In flagella, the central tubules are connected. The central tubules are connected by bridges and is enclosed by the central sheath, which is connected to one of each peripheral doublets by radial spoke. The cilium and flagellum emerge from the centriole-like structures called the basal body. The cilium and flagellum emerge from centriole-like structures called the basal bodies. Centriole is made up of 9 evenly spaced peripheral triplet fibrils of tubulin protein. The central part of proximal region of the centriole is called the hub, which is connected with the tubules of peripheral triplets by radial spokes. The space between two parallel membranes of nuclear envelope is called the perinuclear space. The nuclear matrix is called the nucleoplasm, which contains nucleolus and chromatin. Nucleolus is the site of ribosomal RNA synthesis. The interphase nucleus has a loose and indistinct network of nucleoprotein fibers called chromatin, which contain DNA and some basic proteins called histones. Based on position of centromere, chromosomes are metacentric, submetacentric, acrocentric, and telocentric. The primary constriction of centromere is called centrosome. The primary constriction of chromosome is called centromere. I repeat, the primary constriction of chromosome is called centromere. Sometimes a few centromeres have secondary constriction which gives them the appearance of a small fragment which is called the satellite.